So I think we should be recording now. And like I just said, we'll make a start at this point, but if at any point you want to ask anything, I can pause the recording as well. I actually had a morning session earlier today, and unfortunately, the recording from that didn't work out. The software that we use is a bit of temperamental if there's the slightest of cut in connection or anything. So right when it was recording it, it crashed. So I'm hoping that tonight's recording works out, and that's going to be the version that we'll share with the rest of the class. So if you're looking later on and you're wondering why there's no morning session recording available, that's the reason. And after the morning, I just thought that, okay, I'm not going to do a session by myself pretending to speak to the class when there's no one there. So we'll go with this one recording in this case. As usual, what I really want to do, I want to have a kind of a little bit of an idea. What would be some of the points that I want to cover with you guys tonight? But as always, if anything comes up, even if it's outside the topics that we're covering, just give me a shout out or write to the chat, and we'll address whatever it is. But basically, what I want us to focus on is really on the reviewing the material from the chapter 18. So that was all about the blood, and that's been the content or the focus of our topics this week. I also have put aside some time to talk about next week. So although it feels that it has came super fast, next week we'll do the first exam and also the first practical. I don't want you to worry about it because there's only going to be 2 weeks worth of material in that. So it's going to be really, really easy. But I just want to set your mind at ease by telling what's going to happen and how it's going to map out and what's happening next week. But I want you to know that you're going to get all the help that you need in terms of succeeding on that. So that's just quick walkthrough of what I'm hoping to do tonight. But like I said, at any stage, feel free to give me a shout out and we'll tackle if you've got some questions. I do want to say that you guys are awesome. We're already on to week 3. It feels like we started like yesterday. But if you look that next week, we're going to sit the first set of exams and practicals, or first exam and practical. Once we're done with that week, we're almost 1/4 done on this course, which feels to me like, where has it all gone? So what I want you to know is that the first chapter that we covered, the endocrine system, that was the hardest chapter. It's as hard as it can get. And as we move onwards, all other chapters tend to be a little bit more easier to get a head around because they're concrete things that we can see, we can touch, and if we can see them with naked eye, we can see them at least with a microscope. So do not worry if that felt a little bit vague or a little bit abstract. We're tackling the hardest first, and then it's going to turn easier and easier as we move ahead. And I think that most of the students got all of their assignments in, so I'm very appreciative of that. But I also want to share that if ever you miss out on submitting something in, don't hesitate to contact me. We can always see if something can be arranged, if I can extend your submission time for the assignment, or even if you didn't get a chance to contact me ahead, that can I open something from the past. So I don't want you to feel like you're going to miss the last night's sleep because you're clicking away getting those assignments in right before the deadline comes. But it is a good idea to try to stay on top of those just because the key to succeeding on these online classes is really just getting the work in. There's nothing tougher, there's nothing magical about it than in normal classes. It's just about collecting those little points from everywhere, and then you're all set. So the other thing that came up from few students, and I really appreciate the ones who reached out to me. I always want you to feel that you can let me know if something's working well, or if something's not working, let me know. Sooner I know it, sooner we can figure out how to fix it. So I don't want it to be that at the end of the course, I find out that something that could have been easily changed has been bugging you all this time and making your life miserable. And also, I think that I believe in dialogue. So you let me know, I'll tell you, can I do that, or how can we fix it? But don't hesitate to let me know if there's anything at all where I can help. And I got from a couple of students a message or messages about the fact that they were feeling like, wow, how did this course how does this course have so much homework? That what's going on? It is hard always when you compare online and face-to-face classes, but I just kind of wanted to have a basic idea in my head of how much work we should have. So regardless on which modality you're taking this class, it is 4-credit course. And in a 4-credit course, I think the official definition or official advice that we've been given is, and that's listed in the syllabus, that you should be planning on about 60 hours during the semester in a class and 120 hours doing homework, your independent study, preparing for exams, and all of those things. And if my math serves me correct, that brings us to 180 hours per semester for 4-credit course, which roughly, and I'm really not math major, but it roughly brings us to 10.5 hours per week. So if you're working more than 10 hours on the homework and on the assignments and on stuff, including the time that you spend on actually watching the videos and attending to these sessions, then let me know. Then it's not going the way it should go. And it's not necessarily that you're doing something wrong. It can be that maybe we can find a better way for approaching the study techniques, or maybe we can find a better way of tackling the assignments, or maybe we have to look at, do we have too much homework going on for you guys? So I received a couple of messages of that, and I had a look, and I have to say that I do tend to agree with you. So how this works is that I get the connect assignments from the course lead, and then everything else I do, with the exception of practicals, which have been prepared also by the course lead. So I hadn't really worried. I had gone through them, and I had tried them myself and looked what I'm going to ask from you. But I hadn't really paid attention of how much assignments there was for the week 2 and week 3. So for the endocrine chapter and this blood chapter that we're working on. And I agree that there was a lot. So what I'm going to do, I'm going to go through all of the future chapters, and I'm probably going to adjust so that we have less material to cover or less questions for you in the Learn Smart and also in the quiz, so that they're not so super long with many sub-questions. So I will be adjusting that for every other chapter that comes after the work that we've done so far, because I can't really go backwards if someone has already done all of the work. So week 5 onwards, I'm gonna be doing some changes on those materials in terms of how much homework there is. So I would ask you at this time not to work ahead to week 5 or anywhere beyond that until I send a message that, okay, we've fixed that. So what I really want you to know is that I do care and I do listen, and I want this to be not just busy work, but I want it to be something where you actually learn something. And I'd rather not just do grading. I'd rather interact with you guys and make sure that you're learning rather than just grading faceless tests or exams. So don't hesitate to let me know, and we're going to adjust whatever hasn't been working right now to something that's going to be a little bit better, hopefully, for you guys. And you'll still learn everything, but you won't be doing as much clicking away. Then I wanted to mention few words about this week's assignments. I just think it's always a good idea to highlight if there's anything unusual from past. So we have done in the past the Learn Smarts and quizzes. You're familiar with those. There's nothing new about those. And you've also done the practice anatomy activities. This week, however, we're going to have actually 2 labs that I'm asking you to do. So just don't get surprised by that, and please try to do both of those, because they're again those little points that add up to that bigger stream, which is going to be your final score. It's worthwhile to get those points from the very beginning of the course. And I'm asking you to try to get those submitted by the end of this week. And the reason why I have the practice anatomy highlighted there is just that I remember to mention to you guys, or actually it's the highlights from this morning session, but it helps me to remember that for the practical test that you'll be taking next week, it's really the questions from the practice anatomy activities that you have been doing. So in addition to doing that as a homework and getting those points, that's also you getting set up to succeed on the practical. So that's what I'm asking you to do this week. And if you have any issues at any point with those, with the amount of work, or even with the Connect in general, you just let me know and we'll tackle those. I want to spend the majority of this session to going through the key points from the chapter on blood. But before we do, like I mentioned at the very beginning, I just kind of want to put this on your radar, but I don't want you to worry about it too much. But next week, believe it or not, we're actually already ready to take the exam and practical. And for the entire next week, both of these are going to be open. So you can choose to do it at any time of the week, or you can choose to do one at one time and another one at one time. But the key is that you have to complete, once you start an exam, you have to complete all of it. Or once you start practical, you have to complete that entire practical. Well, we're going to be covering only 2 chapters on each of those. So there's not a lot of material. So I think that that's why it's a nice way to do the first test. So for the exam, which is what I want to focus now, you will be only asked questions from those lecture videos that you've seen. Those are those little videos where we have those PowerPoint slides moving, and I think that I might be in the corner, somewhere at the bottom corner, talking about those. Every question in the exam is from those videos. There's no questions from the labs or anything. Often those topics have overlap, but that's where I would ask you to focus on the lecture video content for the exams. The exam is going to be 50 multiple choice questions. It can be also matching terms or ordering things or things like what you've seen before, very, very similar to that. And you have 60 minutes to complete 50 questions. And if you have had a plan prepared that you might need extra time because of certain accommodations or so on, of course, then your timings or your format might be different. But by default, it's going to be 60 minutes. Do remember that all of my exams and all of my practicals are open book. So you can use any notes that you have. You can use your textbook. You can do whatever helps you to succeed. And you can take it from anywhere where you want. So you can choose yourself. You don't need to have that camera on, and you don't need to be showing the paper like in practical exam that what notes you are taking and so. I also have in most of the exams that I give an extra credit question. So it is in theory possible, and this happens every now and then, that I have students who get more than 100% for their exam score. And that's a good place to be, because any of those extra points carry over to your final grade. So I have had students who have gone to the final exam, for example, knowing that they only need to get half of the points and they've got an A. So I'm just putting it out there that those extra credit questions, if you have extra time and if you're able to tackle that, it's definitely worth doing it. And tonight, whether we're able to talk about it in person or whether it's just going to be the recording that's going to be uploaded, I'm actually planning to tell you what the extra credit question is going to be. So you can prepare on that. And if you prepare ahead of the time, you actually don't have to spend any time of that 60 minutes on that, because it is going to be short answer text about a paragraph, couple of maybe 4 or 5 sentences, and you can write it ahead of the time on a notepad and then upload it during the exam if you wish. So we'll have a look at that a little later. And if I haven't told you what would be the extra credit question by the end of tonight, let me know. I will. And one last thing that I wanted to mention about the exam that we're going to take, on every time that we're taking exams, I'm offering on those weeks 4 review sessions. I have review sessions on Mondays and Wednesdays, and I have on both days 10 o'clock a session, which is the morning session, and then we have this evening session at 8 p.m. It doesn't mean that you have to attend to all of those. The content is going to be the same in all of those. Sometimes the content is formed by what questions students ask, but on average, it's usually fairly similar. If you don't have a chance, you don't even have to attend in person or live to these at all. You can just focus on the recordings. But I am going to be available for you whenever there's an exam week or week when we're taking exams. I'm going to be available on 4 occasions by default, and then any other sessions that you might organize with me individually. So just notice that if that's something that you feel that will work for you. And I remember that I haven't yet uploaded the Monday sessions to the calendar, but they will appear there very soon. So how does the exam then work? Like I said, I will tell you next week all about it. We'll spend all that time getting you ready to take the exam, but to give you a very brief version of that, there is going to be a study guide that I provide you. It's actually already uploaded there, but that study guide is missing some of the key words, or if there's something that I really want you to know, I usually ask you to fill it into the study guide. And some of those blanks are fairly abstract that it's hard to figure out that, okay, what should I write there? Well, that's because you will see answers to those study guides or to that study guide only during these review sessions. So either in live setting or if you are watching them from recording. So I will be giving you every answer to every gap on those. And every question in the exam is from that study guide. So again, since it's an open book test, it might be a good idea to get that completed to guide you, but I'll leave it up to you. About the practical, just a couple of things that I wanted to mention. Again, it's open for the whole week, and all of the material from that practical is going to be from those practice anatomy activities that you've done. So there's going to be the endocrine and there's going to be blood chapter. And really, if you think, what could the questions be? They are the same questions that you've seen those pictures and dragged on those labels on place. However, the big difference on that is that rather than dragging labels to a picture, there's actually going to be an empty spot where you will be typing the answer. So underneath that highlight that kind of came out dark, it says filling the blank. So I don't want you to worry about that. It's not going to be that I would be judging your spelling. I spell horribly, and as long as it sounds roughly correct, you will get the full points. And since there's so little material on the 2 first chapters, the endocrine and blood chapter, there's only going to be 25 questions. And you have 30 minutes to complete those. So it's again roughly about a minute per question. Usually practicals, anatomy practicals are things that you either know it or you don't know it. So you don't really need to sit around thinking about it. However, if you don't know it, Always answer something. I give extra points or half of the points of the question just for telling me about something else about the structure that I'm asking, or telling what it does if you don't know the name of it, or telling something else that's on that area, identifying another important structure. And again, remember, it's going to be open book. You can take it from home. There's no proctoring. So some of the students have found it really useful to watch or to play through that practice practical that I have on week 4 module time and time again, because questions to that practice version are pulled from a larger pool. And if you play through it multiple times, you actually end up seeing every single question that I might ask or that you might be asked in that practical. And as a heads up, I would say that yes, we do have some organs that you might identify based on the endocrine chapter, but a lot is going to be also microscopic images. So that gives you an idea of what sort of things might come. One final thing about the practical, which happens in every course, since you will be filling in your answers by typing in an answer to the empty box, the Canvas tries to grade the activity for you. It doesn't usually do a great job. If there's a spelling error or you use different formatting than what it uses, initially it looks like you wouldn't have gotten great grade. But I will go back to your answers and I will manually grade each one of those. So when you first get the immediate score from the Canvas, do not feel upset and don't feel that that's necessarily your final grade. The final grade, once I've graded all of these practicals manually, I will be sending an announcement of that, and that's the final grade. So just don't let that upset you. So I think that that's all I have on mind about the practical and the exam. Do you have any questions at this point, or are we kind of good to keep going on? Well, in case I don't hear anything, I'm just gonna continue. So I try to keep a record roughly who's been taking part on the sessions. That helps me if your grade is at the halfway point of a move between, say, A and B, and I see that you've always taken part and you're really active. That helps me to give reason to then bring you up. So that's the only thing that I'm writing down. I'm glad to hear that you're doing good. Excellent. So what we're going to do next, we're actually going to go back to this chapter 18 key points, and during the next half an hour or so, what we're going to do, we're going to look at the important parts of this chapter. Of course, there's more to that chapter, and of course you can cover a lot, and there's more finer details, but I want to give you the big points that you need to focus on, so you have a fairly good idea of what have we been talking about. So in case for some reason you wouldn't have a chance to review everything, at least you know the main points. And let me just set up another presentation. And this as well is from the morning, so there's a little bit of highlights already, but I think we can make it work. So you should be seeing a slide where I have all of the organ systems of the human body listed there. And what I'm hoping to do is have you to focus on the cardiovascular system. And this chapter that we're looking this week is all about the blood. So, sorry, I'm just going to put the pen on. So this chapter is going to be all about the blood. And that's the blood and the endocrine system are the only chapters that are going to be in this first exam. However, after the first exam, we'll continue talking about the heart, and then we'll talk about blood vessels. And these 3 chapters put together give us a full appreciation, a full view of what happens in the cardiovascular system. So you'll see if we dedicate 3 weeks to this system, it's probably going to be quite an important one for you to be familiar with. So when we talk about the blood, it's sometimes hard to think that, okay, how do I define what's the function of the blood? What does blood do for us? Why do we need to have blood? And there's many answers to that. But if you need to just pick one answer, I would probably highlight that blood plays important role as the internal transport system. So without this transport system, we wouldn't be able to have many important life-sustaining materials passing from one part of the body to another. So we started by talking about the endocrine system. We looked at the hormones. They all travel in the bloodstream. Same as we will talk later on about oxygen, how your body tissues need that, and you get the oxygen from the lungs, but from there it gets picked up by the blood and transported wherever it's needed. In addition to that, when your cells work, do cellular respiration, do their job, you are left with carbon dioxide as a waste product. Well, blood picks that up from the tissues and transports it to the lungs, where we can get rid of it. Same goes for the waste products. So blood picks up all the waste material from these tissues that do cellular respiration and then transports them wherever we might try to get rid of them, for example, through urinary system or some through respiration and so on and so on. Let's see what else we can find for the functions of blood. So we had a look of the transport function, and I think we covered the hormones, we covered the nutrients, the waste products, oxygen and carbon dioxide. So we covered a good amount of things that blood helps us to transport. And if you had to pick one function for the blood, the transport would be the one that I would emphasize. However, there's other things that the blood also helps us with. And the second one that I've listed there is regulation. So blood helps us to regulate, for example, how much fluids do we have in different parts of the body. So if we have too much somewhere, blood helps us to carry some of that away, or the other way around. Because blood at large is fluid, and it is a great way of transporting extra fluids to other places. Another thing that blood does and helps us to regulate is heat. So if you think when you get really warm, and I think that that's appropriate here in Arizona, you need to get rid of the heat somehow. And usually it is your core, the vital organs, that warm up. Say you go exercising to the gym on a hot day and you're pushing yourself, your body temperature starts to actually rise. But it doesn't rise very high because blood helps you to get rid of some of that heat. That heat is carried to the extremities, to the skin areas, and there you start sweating and you get rid of some of that heat. It works the other way around as well. If we have, for example, your hand, you put it into a cold ice water packet, it will feel cold and it will get cold initially, but then warm blood starts rushing into there to bring up the temperature so it's not quite as cold and those tissues survive that. Same goes for the pH. Our pH in the human body is maintained amazingly within a narrow range, approximately 7.35 to 7.45. If you go much further, you get a little ill, and if you go any further from that, you actually die. So you cannot eat fruit to change your pH. It doesn't work like that. Your pH will always remain as what it is. All of these diets that you see, they're actually more of the things with a certain pH in the food are just good foods for you to eat. You're not changing your body pH. So if we think, what does all this regulation remind us of? Well, we end up noticing that the very same topic that I wanted you to notice at the first chapter, homeostasis, comes up again. So what homeostasis does, we want to keep the internal conditions in your body within a very narrow range and not deviate away from those. Well, that's exactly what we can do with the blood and regulatory functions of the blood. And final one that I mentioned here is the protective function of the blood. So immune system relies on the blood, especially the white blood cells. And we'll talk about white blood cells in a few moments' time a little more. And in addition to that, of course, blood clotting. If we wouldn't form a scab when we get cut, we would just bleed and bleed and bleed to death. That would not be great. So that's another amazing thing that blood plays a role, is protecting us from the extreme blood loss. So those are all functions, and I think that it's always good to start by thinking of these topics and subjects that why does it matter? Well, clearly blood is an important thing for us to consider. The other thing that comes up every now and then is that I sit down with students and I talk about, okay, we have learned these basic tissue types: epithelial tissues, connective tissues, muscle tissues, and nervous tissues. But which one of these is a good fit for blood? And sometimes some students struggle a little bit with this. So that's why I want to highlight this for you. Blood is a connective tissue. And at large, when we discussed about connective tissues, we considered things like tendons, ligaments, bones, all these things that are very concrete and physically connect one thing to another. Well, blood, although it's not as tangible thing as these other ones that we had a look of, blood is still a connective tissue. And how you might think of it is that, well, blood helps us to connect all these things that we had a look of earlier on. So we saw that blood worked as a transport media. Well, that is connecting these sources and destinations of these materials that we had a look here. So remember just that blood is connective tissue, just a special type of connective tissue. And I think that that would make a very good question for the exam. So just saying. Now, I think we're ready to look at the blood and actually look at the elements that make up the blood. But before we do that, I want to discuss how do we end up with this kind of a test tube that you're seeing here. So we've all had, or we probably know someone who has had, or we've seen blood samples taken from us. And they draw the blood, and what you see in the test tube doesn't look like what you're seeing here. What you see is all red stuff. So something must have happened between that blood sample and what you're seeing now. And what happens is actually that sample gets put into a centrifuge, it spins, and that spinning separates different components of the blood to different parts of the tube. And that's because they have a different kind of a weight and they get organized based on that on the test tube. But this is not that bad of a test. This is actually one of the most useful, simple tests that we can do in clinical practice. And it's conducted pretty much everywhere around the world. And already from this simple test, we can get a lot of information about what's going on. So let's have a look what we've got here. So first of all, we'll notice that this sample is divided roughly 50-50 to something that's red and something that's kind of this yellowish, clearish liquid. Well, it's actually going to be 55%, but roughly 50% of this pale, yellowish, straw-colored, a little stickier than water liquid. And this liquid is going to be plasma. So we'll talk about plasma as the very first thing in just a second. But I want you to know that that's the most numerous element of the blood. And then what we see for the rest of this sample, well, we'll see this big red portion, and that's about 45%. So again, close to 50%. And that's red blood cells. And the proper name for red blood cells is erythrocytes. And because I'm kind of talking a lot and I'm making a lot of notes, and it's not just me, but others as well, writing out erythrocytes or writing out red blood cells tends to take a long time. So we often abbreviate it simply as RBCs. So don't wonder if I don't write it out and I just write RBC, I'm talking about red blood cells. And you should be at least able to tell apart the scientific names that we have for these different kinds of cells. So for the red blood cells, remember erythrocytes. Well, that was the 45%, that's kind of the reddish area on this test tube. But you'll notice that in between these two, between the plasma and between red blood cells, we'll see this thin layer, kind of a paler of a material. And sometimes it's described as this buffy gold. That is made of the two other materials that belong to the formed elements. So what we see there is going to be white blood cells. And again, remember, rather than writing white blood cells, I prefer to write WBC. And the scientific name for those is the leukocytes. And then we have also platelets. And both of these are so small in amounts that each one of them is less than 1% of the volume of the blood, of that blood sample. So there's very small amounts of both of these, yet both of those play an important role. And the final thing that I want to mention about this sample that we've been looking, if you look, all of this plasma was this liquidy material. And then we had the red blood cells and also the white blood cells and platelets. They together are called as formed elements of the blood. That's because there's something concrete that we can actually study, we can look at them, we can take microscopic sample, or we can prepare microscopic samples of those. They're not just liquid. There's something concrete. There's actual cells, or at least cell fragments. We don't see cells or cell fragments making up plasma. All of these cells that we see, they're just diluted into this plasma in a normal situation. But just know this term, formed elements. So these three are part of the formed elements: red blood cells, white blood cells, and platelets. So I think as the very first thing that I want to discuss about this is actually, before looking at the plasma, I think we should look at this microscopic image of a blood sample. And this is a very accurate view of what you end up seeing most of the time. And forgive me for those scribbles all over there. That's from the morning. But what we see there is actually all these four things that we just discussed about. So when you have a blood sample, and this blood sample that we're looking is not spinned, so it doesn't look like this. It's not separated one element or one material from another. It's all red. When you take a sample that's all just not centrifuged sample of the blood, and you take a drop of that, you smear it on the microscopy slide, you do some staining. This is going to be H&E staining, one of the most basic ones that we do. You probably put a cover slip on that, and then you stick it underneath the microscope. This is what you end up seeing. So let's see what we can see here. So we can see a lot of cells, those pale reddish structures. We can see also some darker bluish structures, and we can see some very tiny structures there. But they are all, in between them, there's empty space. And all of this empty space is the plasma, or where the plasma would have been. So you'll see it's about half of what we have in this vision if you were to actually measure the area that has been dedicated to different things on there. About half of it is going to be this white, or sometimes if you look on microscope, and especially on older sample, kind of a yellowish, slightly yellowish area. This is the plasma. So plasma is simply this liquid where everything else is embedded. What else can we see? Well, we can see a lot of these red dots. They're quite large, you can tell them apart, but they're not quite as big as some of the ones that we see, the odd ones with the darker center part. So these red dots that we see everywhere, these are going to be our red blood cells. And when you look at them on a microscope, you can almost start to see their shape. And we'll discuss about their shape again in a few moments' time. What else can we see? Well, we can see these larger than red blood cells, and these cells have also this darkly stained nucleus. These are going to be our white blood cells. So we've covered now the plasma, red blood cells, and white blood cells. There's still one more thing that we can see here, but that's going to be a tough one to see. If you look very closely, you might see tiny dots here and there. And I'm going to try. I'm not going to promise that this is going to work. I should be able to zoom in. Yeah. So what we see here is these tiny dots. They're smaller than anything else that we have seen in this sample so far. These are going to be platelets. And platelets are actually just fragments of cells. So they're not full cells on their own. So if we move on to this slide, we end up seeing that, yes, we were right about identifying each of these. And again, we see that the ratio of them in a microscopy slide is very much the same as it would be in that sample that gets spun. So that's kind of the big idea, and rest of this session, which is going to be about 15 to 20 minutes left, we're just simply going to go through each of these. We're going to have a look at plasma, we're going to have a look of formed elements, which are the red blood cells, white blood cells, and platelets. Before I jump into those, I'm just going to stop for a second. If anyone has any questions, you can shout out, but if it's quiet, then we're just going to continue. But don't hesitate to also, you don't have to wait until I ask for questions. Well, it sounds quiet to me. Okay, I love that. All good. Excellent. I try to keep things simple. You'll notice that my exams are simple. I'm a simple guy, and keep it simple. That gets you far in life. Don't complicate things. So let's start with the plasma. So like I said, plasma, if you had to describe it somehow, and I don't know how many of you have had a chance to actually pick up a sample of plasma, but you end up seeing that if you work in the healthcare environment, and you'll notice it is really this slightly yellowish, still somewhat clearish liquid. And like I said, it is kind of more stickier than water, but it's not like glue. Well, why do we have plasma? Well, plasma gave us this volume where all of these cells, all of these formed elements were floating. In addition, plasma is great because it doesn't depend from your blood group that can you get plasma or not. If we give red blood cells, we need to match that you can give only certain kind of red blood cells to only certain kind of recipients. So the donor-recipient relationship matters on that, but it does not matter with plasma. So what we end up doing often is if someone loses a lot of blood all of a sudden, and we want to bring up the volume of the blood as the first thing, and then deal with everything else, we use plasma instead. And what we find in the plasma, well, it's not just a liquid. It actually does something else. We end up finding one of the important things that we find is that plasma contains about 8% of it proteins. And some of the important proteins that you probably have heard about or will hear about are albumin and globulin. I've also listed there the fibrinogen. But in addition to that, plasma actually carries stuff on its own as well. So plasma does carry some of the nutrients, some of the gases, some of the hormones, some of the waste products, and also some inorganic ions. But not quite as efficiently as some of the specialized cells that we'll have a look, but plasma does still do some of that. So I think that that's all I have to say about the plasma. We'll keep it simple. Next thing that we're going to do for the remaining of our time together tonight, we're going to go through these formed elements. So I just want to remind you that formed elements contains the red blood cell and the white blood cells and the platelets. And these are either the red area, about 45%, or the white buffy coat on this sample that we have centrifuged so we get the sample elements of this sample to separate. So all of those three are part of the formed elements. And we're going to go through them in order of starting with the red blood cells, then looking at white blood cells, and finishing off with platelets. So let's have a look of one more general concept before we dive into the red blood cells. So there are certain terms that you end up noticing as part of this chapter that we keep using. And actually, if you learn one term, it's going to make the next term easier for you to learn. So when we are talking about production of the blood, and we're not worrying about what kind of cells of the blood, but blood in general, the scientific terminology or medical terminology that we use to describe that is the process of hematopoiesis. The term hemato refers to blood and poiesis refers to making of something. And when we are going to have a look at what do we call red blood cell production, what do we call white blood cell production, you'll notice that the term poiesis is always part of that. And the first part just changes based on what type of cells we're talking. But in this case, when we're just using a term hematopoiesis, we're talking about the blood in general. And blood is produced in the bone marrow, and then there's specialization processes. But that's why you might see sometimes individuals who have blood-related cancers, for example, they might get a bone marrow transplant and so on. Leukemia would be a good example of that. Because they don't have their own bone marrow, they need some bone marrow to be able to do those blood cells. So we just simply try to move that from one individual who's healthy to them. Okay, now we get to the red blood cells. And it looks a little busy, but it looks busy because I've been scribbling all over it in the morning. So what I just want you to remember that the proper term for red blood cells, the scientific term that we should be using is gonna be erythrocyte. So that's the term that we use when we talk about red blood cells. That would be something that's worth making a note for you. And therefore, if the red blood cells were called erythrocytes, the production of the red blood cells is known as erythropoiesis. So you'll see that term poiesis still hangs around there. And we simply change the first part to identify that now we're not talking about general blood, we're talking especially about the red blood cells. So that's why learning the term erythrocyte is going to help you. And if we look at what do red blood cells look like, you'll often see something that reminds of a kind of a disc that has been flattened from the middle. If I try to think, how could you make that? Maybe if you blow up a balloon of air and then you squash it from the middle, not break it, it would look something like that. Other way to think about it is that it looks a little bit like a donut, but the hole hasn't been fully punched through. It's just thinner in the middle. And actually, if you have looked at the video, or if you will look at the video about the blood typing, what sort of a recipient and what sort of a donor we need to have in this chapter, I actually explain it through donuts and sprinkles that you put on top of the donuts. So if you watch that, you have a good excuse to eat some donuts while you're doing that. So red blood cells are these discs that are flattened from the middle. Fancy wording could be biconcave discs, but we end up finding that they don't have a nucleus in them. And now you're going to be asking me that, how can that be? Every cell that I know of has a nucleus. Well, red blood cell has a nucleus initially, but then it is no longer there as part of the maturation process. We also don't have cell organelles in the red blood cell. And this makes sense if you think of the function of the red blood cell, which is largely to transport gases. If there was a lot of cell organelles, it would probably end up using all of that oxygen that it's trying to transport. In addition to that, we end up finding certain things in the red blood cells. And one of the things that I want to highlight here is that we have 4 hemoglobin subunits. in each red blood cell. And what hemoglobin does and why it's going to be so important for us to have a look of is that that's the part of the red blood cell that actually transports the oxygen. So having those 4 hemoglobin subunits available in a red blood cell is what allows us to do this transport, which was part of that very, very important, if we thought what was the function of the blood, transportation of gases. So what can happen sometimes is that let's consider if we don't have those hemoglobin subunits available. Well, good example of that would be carbon monoxide. So not dioxide, but monoxide. You can get that, from example, from badly burning substances. You can have carbon monoxide. Poisoning, if you have a fireplace and the fire doesn't go out and you close it and it spreads everywhere. Actually, cigarette smoke contains a little bit of carbon monoxide. And what carbon monoxide does, and other gases as well that work in the same way, they actually bind to these hemoglobin subunits. They might not bind into everyone, but they bind to some of those. And as a result, that space that's normally available for the oxygen to go into and be transported in, that is no longer available. So this individual would not be able to transport that much oxygen. So I guess that the moral of the story is that I'm trying to tell you, do not smoke. I don't know. So, few more things that I want to highlight here about the red blood cells. Often when you look at the textbooks, you end up seeing that when we talk about the flow of blood, especially, that there's red arrows and there's blue arrows. And it's almost like it's be telling us that there's red blood and there's blue blood in the body. Well, of course we know there's no blue blood. One type of blood is, though, slightly brighter red. And another type of blood is a little bit more, has a tone of that bluishness. So you can tell them apart, but it's not quite as radical as what you would see normally. And what we want to notice here is that these bright red blood cells, bright red blood cells, actually are the cells that are usually coming from the lungs. So they are full of oxygen. They are very oxygen rich. So they're ideal for going to places where oxygen might be needed. And the term that we use to describe that, or those kinds, is the oxyhemoglobin. That hemoglobin carries oxygen. So there is oxygen in this type of a blood. The opposite of that, the blood that often is the blood that returns to the heart from rest of the body, it does not have oxygen. It has already given off the oxygen from it, so it's gonna be low in oxygen concentration. And if you remember the term D was always an opposite of something. So in this case, it's gonna be deoxyhemoglobin. There's not a lot of oxygen in that. So those are the two that I'd like you to know. And this is really a little bit going on the side of the topic, but of course we lose some red blood cells. Eventually they get eliminated from the blood flow or bloodstream when they get too old, but we have to make some as well. So we kind of try to get to the point where the production and destruction of these is at the same amount. And this slide I wanted to highlight because we talked about the hormones in last chapter. And one hormone that's very relevant here is going to be EPO, so erythropoietin. And what erythropoietin does, you get erythropoietin produced in response to low amount of oxygen in your blood. So what erythropoietin does then, it promotes the production of the red blood cells. So if someone, for example, doesn't get sufficient amount of iron, or they're training in high altitude, or they might have certain medical conditions, there's many reasons why just by naturally you don't have all of a sudden quite as much oxygen in your blood. Well, your body responds by starting to produce erythropoietin. And this is actually why we find that a lot of athletes that are competing on a high level, before they go into a competition, they actually go to train on a high altitude camp. And the point of that, or the idea of that, is that, okay, in high altitude, there is overall less oxygen available. So the body will perceive that there's very little oxygen in the blood, and body will start producing this erythropoietin, which then increases the amounts of red blood cells. And all of the sudden, when these individuals come to the competition site, which is not in high altitude, now they have a lot of extra red blood cells, which allows them to carry extra oxygen to their muscles. So theoretically, they're able to compete or able to do more muscle activity than their peers. This is allowed. This is okay. This is not cheating. However, what happens sometimes is that some athletes take synthetic erythropoietin and inject it into themselves so that their body will start producing red blood cells. That is doping. That is not allowed. So you'll see that these systems start to slowly overlap in terms of what we have a look. And really what I wanted to highlight with this slide is not all this other information that's there, but yes, we talk about anemia when there's low concentration of oxygen in blood, but response to that was, as we learned, the EPO, hormone erythropoietin, which is produced from the liver and from the kidneys. So that's all that why I had this slide. I just do them so I remember to talk to you about everything. And the last thing before I kind of jump into what's the extra credit question is going to be that how long do we have red blood cells in our body? How often do they need to renew? Somewhere around 100, 120 days is the typical lifespan of the red blood cell. And what we do with red blood cells when we remove them from the bloodstream, we're not going to waste them. We're going to break them apart and take away the important elements of those that we can reuse. So we will be removing the heme groups, we will be removing iron, and we will be removing the globin groups as well. So hemoglobin is broken into heme and globin. And these can be reused in the process of building new red blood cells. So I know that has been a lot. Do we feel okay still? I think I'm not hearing at least anyone being against. Okay, good. This slide is busy because I scribbled all over it in the morning. I'm glad to hear it. Excellent. I promise that we won't run too much over. I know that we're right on the edge, but this is probably the last big topic that I want to mention. So when we look at red blood cells, we saw that normally they look like these flattened discs that you can see at the top right corner of this picture. That's a normal, healthy red blood cell. But if we have one base on the DNA that's coded wrongly, instead of an amino acid that's then produced from that DNA, it's something else than it should be. And this is just one single base and one single amino acid. The red blood cell changes its shape completely. What we get is a sickle cell disease. So sickled cells look a little bit like these half moons or crescent-shaped red blood cells. Well, if we think, why would that be a problem? And why would it be good to be a shape of a normal red blood cell? Let's consider how red blood cells have to squeeze through smallest of blood vessels and smallest of capillaries in our body. Well, some of those are just enough in terms of their diameter that red blood cell can squeeze through it, having that smooth surface and it gets through that. Well, if you have sickled cell, it might not be able to pass through these vessels that are the smallest. So what we end up finding that people with sickle cells, they end up developing these red blood cells building up in places where it shouldn't be building up. The other thing that we end up finding is that this normal red blood cell was able to transport, we remember, 4 oxygen units at one time. However, if the red blood cell is sickled in shape, it's still able to carry some, but not quite as much. So that's not ideal in that situation. What I want to talk with you, and this is part of the extra credit question. So first of all, that you know what's the difference between the sickle cell disease and normal condition of the red blood cells. And that was, of course, this shape. And what are the consequences of that? Well, we remember that we had more oxygen carried in the normally shaped red blood cells and less oxygen in these sickled cells. The other thing was that they ended up getting clumped into places where they shouldn't get stuck. Well, when we look at a map of Africa, we'll notice that sickle cell anemia, sickle cell diseases in general, not just in Africa, but everywhere in the world, are usually more common in individuals whose ancestors come from sub-Saharan Africa. And especially even in Africa, we'll notice that it is this area underneath the Saharan desert where the sickle cell becomes more and sickle cell diseases become more and more and more common. Well, you would think that that really sucks for these individuals, that they really didn't luck out at all. Well, we might have thought about so for the longest of time, but this is what I think is so cool about sciences. If we take another thing that we end up seeing in sub-Saharan Africa, malaria parasites and malaria diseases, we'll notice that the area where malaria is common almost matches exactly with this area where sickle cell disease is common. So there has to be a reason why it is found in the same areas. Well, what we know is that malaria is actually a parasite that enters to your body, enters into your blood, goes inside the red blood cell and increases there in numbers, then kills that red blood cell and spreads elsewhere. Well, turns out that individuals who have sickled red blood cells, these malaria parasites are not able to go inside. So basically, having a sickle cell disease can actually protect you against malaria and malaria parasites. So that would be a great extra credit question. A, tell me what sickle cell disease is. Well, we remember that sickle cell disease was when there's a single base difference, and for that reason, the red blood cell takes this crescent shape. What is the downside of having sickle cell disease? Well, it doesn't carry as well oxygen, these red blood cells, and they got caught into places where they shouldn't have. What might be positive about having sickle cell disease? Well, we just learned that malaria parasite is not actually able to enter to these sickled cells. So it might be protective towards that issue. And then the last question that I'm going to ask on that is that what does that tell you about evolution? And you don't have to believe in evolution. Basically, that tells that nature always plans things that are in our best interest. Something that's going to help you to succeed better is going to be a characteristic that gets passed onwards to the offspring. So evolution works in a smart way, even when we don't see why something might be beneficial. So that would make a really good extra credit question. That is the extra credit question. I don't want you to spend too much time answering on that. One paragraph is enough. I mentioned those questions, maybe 4 or 5 sentences. You can write it ahead of the time. You have that 60 minutes for 50 questions. The 51st question is going to be extra credit question. If you want, you can write it on the notepad ahead of the time and then just copy paste. You probably spend 5 seconds of your exam time to answer to the extra credit question. And I'm super generous always with the extra credit questions. So just putting it out there. I know that we're a little bit over the time that we had allocated for this session, but I'm going to finish it. If you have to go, I totally get it. I'm going to try to give a really brief overview of what you need to know about white blood cells and platelets. So let's have a look at the white blood cells as the first ones. And the fancy term for the white blood cells is gonna be these leukocytes. So notice how it's again a different term. And the white blood cells on these diagrams, you remember they were bigger than any other cells that we saw in this sample, and their nucleus stained dark. They're actually very low in terms of the numbers in the blood, but they actually do a lot. And the number of white blood cells can increase if you have an infection, for example, if you have pathogen that has invaded your body, if we need to develop an immune response. But on average, in a healthy person, it's about 1% of the blood that's white blood cells. So the function of the white blood cells is really the defense function. They defend you from various diseases, various pathogens, and so on. Well, we're going to look at 5 different kinds of white blood cells. And these are often classified based on whether we see granules in their cytoplasm or if we do not see granules in their cytoplasm. Do you need to be able to write all of these from your memory? For the exam, you don't need to, but for the practical, you should be. So in the practical, I can tell you there's going to be pictures of different white blood cells, and you're asked to identify that what kind of a white blood cell could it be. So let's look really quickly. I know we're over the time, but still I'm going to go through really quickly. How do you identify each one of these? So the neutrophils are going to be the most common white blood cells that you find in the body. About 50 to up to 70% of all of the white blood cells are going to be neutrophils. And if you look at them, well, we see that there's granules in their cytoplasm. Again, I'm trying to zoom in. I make no promises that I know how to do it. It does work. So you'll notice there's little dots that we end up seeing there. So that's the first clue that it's going to be neutrophil. The second clue is going to be that we notice this nucleus that has stained dark, and it's in there in various different lobules that are connected to each other. So it's not just a single ball, it's a group of multiple, and these multiple are connected to each other. If you find those 2 characteristics in that cell, it's going to be neutrophil. The next one that I want to talk with you is going to be the eosinophils. Eosinophils actually play more of a role in protecting you against parasites, whereas the neutrophils were all about protecting or working in your immune defense. When we look at the eosinophils, what can we tell? How do we identify? Well, first of all, we see again, there's those granules in the cytoplasm. And the second characteristic that specifies this is that they are going to be binucleate. So there's going to be nucleus that's present in 2 parts. If you see that, that's going to be eosinophil. So I'm just going to add that there. Well, the third and last type of a white blood cell that has granules is going to be the basophils. Basophils, if you look at the numbers, again, very low in numbers, but they have a role, they're needed. And let's zoom in and have a look. And I think with basophils, you can again see really well those granules. But what you'll also end up seeing is that the nucleus takes all of the cell area. So if you see granules and it's all nucleus, that cell, that's probably going to be basophil. Basophils play a role in inflammation responses. Antihistamines would work to calm down the basophils, so allergic reactions. Eosinophils also have some amount of role in allergic reactions. Well, the 2 ones that we have not looked at yet are going to be these white blood cells that do not have granules in their cytoplasm. So the cytoplasm is smoother. There's not these distinct dots. The first one that we're going to have a look of is going to be lymphocytes. They are second most common white blood cell that we find in our body. And if we look at them, they are they have that smooth cytoplasm, and the nucleus takes place almost entire cell, but not quite. These cells, lymphocytes, stain typically dark. They don't have that purplish or reddish color. And the lymphocytes, they really play a role in terms of the antibodies and immune responses. So when we talk about immune system, it's going to be the T cells, B cells. We're going to be looking at that kind of lymphocytes. And the final one that I want to mention here is going to be the monocytes. Monocytes, again, we see certain characteristics that help us to separate them from others. Smooth cytoplasm, and their nucleus is almost like this horseshoe shaped. And what monocytes do, they work in the phagocytosis. So they eat away pathogens that shouldn't be in your body. They engulf them inside them. So those are the 5 different kinds of white blood cells. For the practical, yes, you want to be able to identify them and you want to be able to write their names. So you don't get the list of names in a practical. You have to type it in. If you do a spelling error, just wait until I've regraded the practical and you'll get points regardless. Initially, the Canvas might be a little bit unhappy about it, but just wait until I regrade it. Well, in the exam, I don't ask you to even remember these words, that which one, how did you spell which one of these. What I will ask you though is to tell me that what of the white blood cells are most common, which are least common, and then the 3 in between those. And you will be given these names of the white blood cells. Well, I have a mnemonic for you. If you organize these white blood cells in the order of how common they are, you can come up with a mnemonic that's never let monkeys eat bananas. If you organize them based on the first letter, that mnemonic tells you the order. Neutrophils being the most common and basophils being the least common. So just remember, never let monkeys eat bananas. There is literally a question. I think I asked you to pick that one in the exam. Well, what happens in terms of terminology? We saw that we talked about leukocytes. So the production of the white blood cells is then going to be leukopoiesis. And again, we remember that they originated from the stem cells. The last, and I promise this is probably going to take 1 minute to finish this off. Last one that we're going to have a look at is going to be platelets. And we saw earlier on that these were these tiniest dots that you could see here and there. Not very common, but here and there in the blood. Again, less than 1% of the blood volume. They are not full cells, they're actually fragments of cells, and they play a role in the clotting of the blood. And platelets stick around only for about 10 days in your body, and then they have to be renewed. So there's a constant process of replacing those and removing those. Well, why do we have platelets? They help us to form a cloth, they help us to form the scab if you cut yourself. It turns out there are people who don't produce at all or enough platelets in their body. And I would happen to be one of those. So if I get a cut, and if I wouldn't take medicine to help me to get synthetic platelets or help with my platelet production, I would just keep bleeding and bleeding and bleeding until I would die. So my blood doesn't contain enough platelets, and that's why it's not able to clot, it's not able to form those scabs. But luckily, medicine helps me out and I'm fine, I can live a normal life. There's many famous individuals who have had this issue. You'll end up seeing if you read, for example, about Russian Tsars and so on. So that completes everything that I wanted to say about the material on this chapter. And I promise I am not going to take a lot of your time. I'm just going to show the final slides that I always show. So that gives you a good overview. And I just want to remind you that, let me open up. Here we go. I just want to quickly remind you that if you are feeling that you could use a little bit of help with studying, there's been a lot going on or a lot to cover, remember you have tutors, and tutoring is free as part of your degree. So since we cannot meet in face-to-face context, our tutors are actually available for you at the Cranium Cafe. You'll end up seeing this, I didn't mean to go backwards. You'll end up seeing this link in many different places. So that's something to bear in mind. If that feels like something that could benefit you, look up the Cranium Cafe. The address is there. It's in the syllabus. It's all over the website. You do not need to have an appointment. You can just show up and you'll see a list of tutors that are available, a little bit like what are their specialties or what they are experts on. And you can pick the one that you like. And if the tutor is available, then you will be just seated with that tutor. You'll have one-on-one session and you can work through whatever you need help with on your school things. If you like a certain tutor and they're already busy, you just get placed into a lobby and you'll wait until they become free. So that's the first way of getting help and tutoring. And the second one is, of course, that I'm here for you. So you can ask me to meet you in virtual setting, one-on-one, or if you have a group of people who are studying together, have a study group, or as a whole class, I'll make time. You just have to let me know that you want help and I will make the time for that. And that brings us to the very end of what I had planned for today. Do remember that next week, both on Mondays and Wednesdays, I have the sessions at 10 and at 8. And you should not worry about the exam. I will set you up to get full points for that. So we'll get you set up for that. But before that, let's wrap up this last chapter of the blood, and you should be all set. If there's any questions, this is a great time to let me know. Otherwise, this is all there is. Thank you, Savannah. I really appreciate you joining. Have a good night. And like I said, I know there's a lot of homework for the 2 first chapters, but I will fix it for the remaining chapters. Okay, have a good one. Thank you for joining, Michael. Excellent. Okay, good night, everyone. Good night. I appreciate you joining. And I'm going to wrap up this recording as well. So if you have any questions and you couldn't join me in live, then you just send me a message. Canvas is the best place. But thank you for joining me today.